Good evening. Let's begin by singing How Great Thou Art. So, how great thou art. O Lord my God, when I in awesome wonder consider all the worlds thy hands have made, I see the stars, I hear the rolling thunder, thy power throughout the universe displayed. Then sings my soul, my Savior God, to thee. How great thou art, how great thou art. Then sings my soul, my Savior God, to thee. How great thou art, how great thou art. When through the woods and forest ways I wander, and hear the birds sing sweetly in the trees, when I look down from lofty mountain grandeur, and hear the brook and feel the gentle breeze, then sings my soul, my Savior God, to thee. How great thou art, how great thou art. Then sings my soul, my Savior God, to thee. How great thou art, how great And when I think that God his Son not sparing, sent him to die, I scarce can take it in, that on the cross my burden gladly bearing, he bled and died to take away my sin. Then sings my soul, my Savior God, to thee. How great thou art, how great thou art. Then sings my soul, my Savior God, to thee. How great thou art, how great thou art. When Christ shall come with shout of acclamation, and take me home, what joy shall fill my heart. Then I shall bow in humble adoration, and there proclaim, my God, how great thou art. Then sings my soul, my Savior God, to thee. How great thou art, how great thou art. Then sings my soul, my Savior God, to thee. How great thou art, how great thou art.
Good evening, everyone. Looks like we have some new faces and a little bit of a bigger crowd. I'm excited this evening to hear what Dr. Carter has for us, and I hope you all are too. Before I get started this evening, I just want to go over a little bit of what he talked about last session, or this morning's session, on the historical Adam. And I really appreciated how you shared and brought that out at the end, especially that verse. It meant a lot to me, and I think it really pointed it towards the Bible, which I really appreciate that about you. Everything that we've learned here, it still points towards the Bible at the end. And that one verse you shared at the end, 1 Peter 3:15, but in your hearts revere Christ as Lord, always being prepared to give an answer to everyone who asks, to give a reason for hope that you have, but to do this with gentleness and respect. And I want you guys to think about this. And I want us to be challenged by this, do you have an answer to everyone who asks about the hope we have? And I think that's what you are sharing and showing us in your topics. So to start off, I'm going to read one of the Psalms. So I want you guys to turn with me to Psalms 19, and we're going to read over the Psalms, this Psalm. The heavens declare the glory of God. The skies proclaim his work. The skies proclaim the work of his hands. Day after day they pour forth speech. Night after night they reveal knowledge. They have no speech. They use no words. No sound is heard in them. Yet their voice goes out into all the earth, their words to the ends of the world. In the heavens, God has pitched a tent for the sun. It is like a bridegroom coming out of his chamber, like a champion rejoicing to run his course. It rises at one end of the heavens and makes its circuit to the other end. Nothing is deprived of its warmth. The rest of the chapter, it goes on to read about some other really good things. But these 6 verses are the ones I want to really think about tonight, and before he gets to his topic. I know we're talking about the cell, which is quite a bit smaller than the sun, and in here it's talking about more the heavens, but I also think this is something we can see in all creation, and I think we can see it just as much in the beauty of the cell. So I'm going to read those 3 verses again. Day after day they pour forth speech. Night after night they reveal knowledge. They have no speech. They use no words. No sound is heard from them. Yet their voice goes out into all the earth, their words to the ends of the world. David understood that way back then, and I hope we can understand that tonight with this presentation of the cell. I have a few announcements before I turn the time over to Dr. Carter. As you guys can see to my right here, there's a nursery. If you guys need that at any time, it's right over here. And we also have restrooms out here in the hallway. Last time we forgot to say something about that. So if you need the restrooms out here in the hallway, you guys can find it there. And yes, also tomorrow morning we have the morning service at 10. I think later Kurt will explain more about that. So without further ado, I'm going to introduce tonight Dr. Carter Williams, which I'm really excited to hear about his topic tonight, as you can see, the high-tech cell. Dr. Carter has spent 4 years teaching high school and biology and chemistry and physics. He obtained a Bachelor in Science and Applied Biology in 1992 from the Georgia Institute of Technology. In 2003, Dr. Carter obtained a PhD from the University of Miami in marine biology with a discretion in a big word that I can't even understand, but it's called nitrogen fluorescent proteins. And yeah, we heard a little bit about that in our last topic. Dr. Carter is currently a senior scientist and speaking for the Creation Ministries International in Atlanta, Georgia, and does research in human genetics and other issues related to the biblical creation. So let's turn the time to him, and I will welcome up here, and then I'll pray for him and let him begin his topic. Lord, I just thank you for your creation and how it speaks without any words, Lord. How we look at it and just in awe of who you are, Lord. Thank you for this man, Dr. Carter, that he can study this and he can present this to us in a way that can just open our minds to you, Lord, and point our faith more in you, Lord. And I pray that this evening we can all be touched and that the Holy Spirit can move. In Jesus' name I pray. Amen.
Good evening, everybody. That's much better. Technology. What are you staring at? That is something that's inside every one of your cells. You have millions of them, millions in each cell. This is the world's smallest motor. It's an electric motor, but it doesn't run on electrons, it runs on protons, hydrogen ions. It takes an incredibly fragile molecule called adenosine diphosphate and joins it to another phosphate to make an incredibly, incredibly fragile molecule called adenosine triphosphate. A warm little pond cannot possibly produce something like this. And let alone that, it can't even produce the adenosine or the adenosine monophosphate or the adenosine diphosphate. And phosphate in nature, if it touches any metal ion, it forms an insoluble salt and precipitates out of solution, which is why phosphate mining across the world is so incredibly important for farmers, because phosphate disappears and gets locked up and then plants die. So our bodies use an incredibly ridiculous system for producing energy. That's the thing that makes energy. I have too much of it, apparently. But I'm going to take a little detour here because I want to talk about what we've been doing all day outside. There's a machine out there on a table. That machine measures the angle to the sun up and the angle around from north to the sun. It's called the azimuth and the altitude. Well, I got on the NOAA website today, that's the National Oceanic and Atmospheric Administration, and I opened up their solar position calculator. I typed in the latitude and longitude of this location, and I adjusted the time. It's okay, give me the sun's position at 7 o'clock, 8 o'clock, 9 o'clock, 10 o'clock, and 11 o'clock, all day long. And this is the results I got. So in red, that's the angle from north. And you can see it speeds up a little bit and slows down and speeds up again. And in blue, that's the angle above the horizon of the sun. It gets up to about 52 degrees above the horizon. That's your latitude. In June, on June 21st, oh, by the way, how many of you light-skinned European types got a nice sunburn today? We're 2 months away from the maximum height of the sun in the sky in the middle of June. Well, 2 months after that is August. This is the sun of August. And you thought it was still winter. Just saying. I hope you can sleep tonight. And I got roasted too, but not as bad as some of you. Oh boy. Okay, so now we're learning about the sun in science. That is a mathematical model based on the assumption that the earth is a sphere and the sun goes around the earth. The flat earth community believes basically that Noah is the heart of the beast, and they do nothing but lie. That is the model. Now, a flat earth model would look nothing like that. The earth is flat, the sun circles above the sky, it never touches the horizon. And yet, it starts off on the left, on the horizon, on the right, on the horizon. Okay. Now, this is our data sheet. How many of you participated in this? How many of you took a measurement? Okay, look around, everybody. Ready? Let's take a vote. Let's vote on, I did half of these measurements, everyone else did the rest. Let's take a vote. How well do you think we did? There's three options. Terribly. Our dots are going to be all over the place, have nothing to do with that. That's option one. Option two is pretty good, but our dots would be like, but follow the line, kind of. And the third option is we nailed it, using this medieval torture device out there. Okay, let's vote. Everyone, you have to vote. You have to pick one of those three. We're going to go in order. Who thinks terribly there's no relation between our data points and that? Raise your hands. Okay, someone who has no confidence in me. 2 people, no confidence in me, okay. How about option B? Kind of following the line, but the points should be all over the place. Oh, okay. Now, how many of you actually think we did it correctly and our points are going to follow the line? Are you kidding me? Wow, you're very confident people. Because I didn't expect that. I didn't think that machine was going to be accurate. Here are our results. My friends, welcome to science. Also, if someone wants to claim that the earth is flat, they have to call all those people who had their hands up liars. And all of you who witnessed this. We did a public social experiment. If you'd like to take a photograph of the data sheet and put it in Excel and do your own calculations, it's there. Anyone can access it. Now, I'm not talking about this tonight. That's Monday morning. The shape of the earth is really important, and it's really fun. And it gets really esoteric. But we nailed it because the earth actually is a sphere, and the sun really does go around the earth. And there's no conspiracy. Okay. Now, we're going to get into the high-tech cell. I'm going to, of course, promote Creation Magazine because it's a very critical thing in my life. I think I'm a Christian because of Creation Magazine. I mean, I was a freshman, maybe a sophomore at Georgia Tech. I was a complete total nerd, sold out to science. I would have said evolution's a fact, millions of years is true. And I went to hear this creation speaker at a church in Atlanta. And he was saying that the earth isn't millions of years old and evolution isn't true. And I thought that man was crazy. I am now that man. But I went back to my dorm room with a copy of Creation Magazine. And there was an article in there about something I learned in my evolutionary biology class the day before. And I had no idea it could be answered biblically. And there's another article in that edition that had something about geology that I'd always wondered about, and it answered it biblically, and I was shocked. And I think this is the tool that God used to study me into Christianity. Now, I understand we walk by faith. I understand that clearly. But I'm a nerd, and I ask questions, and I don't like when I have a question that can't be answered or that seems to conflict. When I find an answer that conflicts, I go and try to figure it out. And if Christianity and the Bible wasn't true, I shouldn't have gone down this path. I should have proven the Bible wasn't true, and yet here I am. We also have our website. For people who haven't been here before, just give me a second here. Our website, creation.com. There's flat earth stuff's on the website. There's high-tech cell stuff's on the website. All the stuff we talk about on this week is on that website in video form, in podcast form, in article form. We're on all the social media sites you can imagine. We have a place everywhere we can possibly get ourselves. Okay, that's the introduction to our ministry. Let's get into the meat of what we're talking about tonight. We're going to be talking about what's inside the cell. We'll talk a little bit about DNA, which as far as I'm concerned is the most complex information storage mechanism in the known universe. That's my definition of DNA. But we're going to talk a lot about more than just DNA, because there's a lot of amazing things in our cells. But I want to consider something we showed this morning. Just talk about it again very briefly. This is a photograph of the human genome. You have chromosomes in your cell made of DNA. And when your cells divide, those chromosomes condense. You can put a stain under a microscope slide, and you can see them in a microscope. You can cut them out and line them up, and that is your genome. Well, actually, that's a man's genome because there's a Y chromosome on the bottom right. Ladies, you have 2 X chromosomes. And as I said earlier, you have 6 feet of DNA in every one of your little microscopic cells. That tells us already God's a genius, right? Okay. Now, when they went to sequence the human genome, it took them 13 years and $3 billion. That's $1 a letter. And the data wasn't that good. There was gaps, there were mistakes. It was awkward, but that was it. Today, I had to update, I should say, millions of human genomes are now available for public consumption. And the $1,000 genome is already passed. I mean, I had my genome sequenced for $125. That's how fast technology has advanced. From $1 billion or $3 billion to less than $300 in about 20 years. They're now inventing machines that can sequence DNA on the fly. So you could put one in like a bus or a subway or a school, and as our DNA is just floating through the air, it'll suck it in and sequence it. That will allow them to detect viruses, bacteria, or individual people. Now, if you think you're safe because maybe you haven't participated in Ancestry.com or 23andMe.com, no. All you need is a second cousin that can positively identify you from any DNA sample. And as soon as there are about 1 million people involved in some public project, and there are more than that already, you can be identified specifically, individually, unless you have a twin, using it probably costs $1, because you don't have to sequence the whole genome, you only have to look at a couple letters. Now, this genome of ours, it controls things like biochemistry. You ever studied biochemistry? I love it. But there's no end to it. It's really complicated. It goes on forever. We have all these crazy chemicals in our bodies. Chemicals that make a synthetic chemist drool because they can't make them. They can get them from biology, but they actually can't make them in a laboratory because it's too complicated. And these molecules are transformed into other molecules using enzymes, using proteins. Your body produces protein to take one improbable molecule and turn it into another improbable molecule. But it acts like scissors, like a scalpel. It can literally, you have enzymes that can move one atom off or onto a molecule. We can't do that in a laboratory or even in a factory. We do dirty, bulk, mass action chemistry. You throw a bunch of stuff in there and you let it react, and then you try to purify what's left and keep the non-purified stuff out, because usually that stuff is toxins. And you just got to pull out the molecule you want. It's really expensive and really hard, but biology does amazing things. And all that amazement argues against evolution. Think how delicately balanced the system is. One mutation in one letter of one protein, and you're dead or deformed. Now, some of them can withstand a mutation, but usually a protein has an active site, and that can hardly be changed. And if that can hardly be changed, how does it evolve? You know what a genome is, right? That's all your DNA. There's other omes out there. There's the proteome, that's all your proteins. And there's the interactome. That's all the interactions amongst all the proteins in your body. This is a map of the interactome of a fruit fly. But only about 1/3 of the proteins that they mapped are shown, because they wouldn't be able to show them all. That'd be too much. But what this is in green, the proteins that are inside the cell nucleus. In blue, the proteins that are inside the cytoplasm. And yellow are the membrane-bound proteins and extracellular proteins. And you can see there's a cutout here and a zoom up, and another cutout and a zoom up. Each one of these dots is a protein. Can you see the lines connecting them? Well, this one in the middle, I can count 1, 2, 3, 4, 5, 6, looks like 7 or 8 lines connecting to other proteins. But what are my connections? I mean interactions like this. Let's say that this is a protein. There's 8 other proteins that interact with this, but one of them comes and grabs onto it and hides the button so it can't work. One of them comes and pulls out the little dongle and shoves it into my computer. Another one comes and turns it on and off. Another one comes and pushes one of the buttons. That's what we're talking about. We're talking about a lock with multiple keys. What? And if you change those keys, you can't open the lock, or you break the lock, or it gets stuck in the off position, or gets stuck in the on position, and then you die. Some of these things can be changed. You break one of these things, maybe there's an alternate biochemical pathway that can get to the same result. Fine, but it's very inefficient. Our systems are designed to withstand lots of abuse. That's a mark of a good engineer, by the way. Our bodies are robust. As we age and get older and older and older, we're picking up more and more mutations. More and more things are breaking in our bodies, and yet we still keep going till we finally give up. But it's amazing. We can last for 80, 90 years. That's crazy. I could not make a machine or a robot that lasts for 80 or 90 years. There's no way. And yet our bodies do this all the time. When we don't get hit by a bus or eaten by a bear or something, But if you can't change most of those connections in a fruit fly, how did the fruit fly evolve? And if you can't change it, how does it keep on evolving? And the human interactome is at least 10 times more complicated than that of a fruit fly. So I submit this to you. Once you have living things, those living things can change. That's Monday night's talk. Once you have living things, they can be modified. They can be tweaked. Mutations can happen and change the skin color, the hair color, the eye color, the height, the weight. Fine. But those changes don't explain how the thing came into existence in the first place. And that's one of the keys to the evolution creation debate. Everyone says, yeah, things can change, fine. But Mr. Evolutionist, the types of changes we see are not sufficient to explain your theory. They fit beautifully into the creationist ideas. Beautifully. Everything that Charles Darwin wrote, The Origin of Species, I'm like, yep, sure, okay, fine. And after about 150 pages or something, he goes, I can see no limit to the amount of change that natural selection can produce. Oh, you can see no limit. You mean in your imagination? Yeah, in his imagination. There was actually no mechanism to do it. You right so far? All right. I submit to you that the genome is not simple. It is the most complex computer operating system in existence. But it's not static. It actually operates in 4 dimensions. 4 dimensions. What does that mean? You remember in math class? A point has no dimensions, right? A point has no height, width, or length. But a line has 1 dimension, length. Remember that? Okay, a plane has 2 dimensions, length and width. You tracking with me? What grade was this? 7th grade, 8th grade? It was a long time ago for some of you. But a cube has length, width, and height. Those are the 3 spatial dimensions. What's the 4th dimension? Time. Your genome operates on all 4 of those dimensions. Let's go through them one at a time. 1, the first dimension is the linear string of letters. I said you have 6 feet of DNA in every cell, but it's only a few nanometers thick. It is literally a line. It has almost no thickness and is 6 feet long. That's crazy. But if you look at that information, like here, the beginning of the Y chromosome, man, that's boring. And it's highly repetitive. TCCC, 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 TCCC. I just picked a spot in the middle there. It's highly repetitive, super boring, and the human brain cannot grasp this type of information. You could try to read through the genome. It's only 3 billion letters, only take you 30 years or something like that, but you just go insane if you tried. But let's change this up. Let's say a different way to visualize this. Let's replace each of those letters with a dot. And we'll color them. A, C, G, and T become yellow, green, red, blue, and yellow. I don't remember the 4 colors are, but here they come. Same exact information in color. And your brain instantly picks up patterns, doesn't it? Yeah, why? Because we're pattern recognition machines. And the human brain is shockingly good at it. Can you see this pattern right here? It's a repeat. The repeat is this long. And then the next line is the same repeat. And every once in a while, there's a break in the repeat because there's a little deletion. And all of a sudden, it stutters and it goes a little bit. That's very typical in high repetitive DNA. And what's this big gray spot? That's a place in the genome that the original Human Genome Project couldn't sequence. They left 50,000 letters, 100,000 letters, 10,000 letters, big gaps, because they said, okay, we know that there's something in there, but we can't sequence it because it's too repetitive, and the sequencing machines only sequence that at that time about 300 letters at a time. So you find 300 letters, where does it go in this repeat? It could fit here, 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 here. You don't know. Just like you get in your car, and you turn the radio on, and your favorite song is playing. And you start singing along, and all of a sudden, oh, whoops, you realize it's the end of the song, or the middle of the song, and you start singing the wrong verse, right? because your sample wasn't long enough to know where you belong in the song, and the song has a lot of repetition in it. Same thing here. The early machines cannot do this. Now, last summer, 21 years after the human genome was complete, they finally finished the human genome. And very soon they came out the chimpanzee, the gorilla, the orangutan, and a monkey within months. So we now have, for the first time ever, complete genomes. But look, down here, there's a different repeat of a different length. There is high order information in the genome. We don't know what this stuff is for. The evolutionists have consistently just called this junk. Wait. Let's look at the second dimension of the genome. And what's that? It's the long-distance DNA interactions. You have this piece of DNA makes a protein that comes over here and turns something on or turns something off. The classic example is a sugar digesting system in E. coli. It's called a lac operon. Lac is for lactose. When there's glucose, sugar, in the medium, the bacteria doesn't try to digest lactose, milk sugar, because it's energy inefficient. But if there's no glucose and there's lactose present, the bacteria is like, well, why not just go eat that lactose? It's right there. So it turns on this whole suite of genes. There's actually 3 of them in a row: LAC-Z, LAC-Y, and LAC-A. There's some things above the gene at the beginning of it that tell it whether it can turn on or off. There's a repressor protein. If glucose is around, the cell makes a repressor protein so the LAC genes don't turn on. If there's no glucose, that is gone, and a cap protein is produced that'll stick here, then RNA polymerase can zip down the thing and manufacture this proteins so that it can digest lactose. You understand? This is an easy example. This was figured out in the 60s. And because it was figured out in the 60s, the evolutionists came up with this thing called, actually it's from the 40s, the one gene, one enzyme hypothesis. You have a protein gene makes a protein. And life seems so simple. And the rest of the 98% of our genomes that don't code for protein, they're just junk. And that was a ruling paradigm for about 30 years. It was always wrong, and they always knew it, but they fought tooth and nail, and even now they're still fighting to resist any evidence of function in that junk DNA stuff. But if you were to map out for the human genome, the second dimension, you'd have to write all the DNA in lines and then draw arrows from one place to another where one part of a DNA interacts with another part. In fact, if you wanted to do that, you'd see nothing but red arrows. There'd be so many interactions. That's the second dimension. See that? You have to do it in an XY plane, height and width, to draw it out. You get that, why that's two-dimensional? Okay. Oh, there's more. MicroRNA. Oh, junk DNA. Not junk. These are all these small RNAs that are produced that interact to control atherosclerosis, which is hardening of the arteries. That's a very complicated system that is very liable to mutational breakage. And if you break that system, you die of a heart attack. This is why we eat healthy, my friends. This is why we avoid trans fats, my friends. Just saying. But man, I know pork skins are good, but I ain't eating them. Anyway, just saying. It's not the pork, it's what it's fried in. We can also compare the genome to a computer program. But when we do, we realize that computers are very different than biology. On the left, transcription is when a cell turns DNA into RNA. And some of that RNA will be translated to protein, but transcription is the first step. Not all the RNAs are made into proteins, but it is taking the DNA and turning it to RNA. When we look at E. coli, we can see that there are some master level programs that control about the same number of middle level programs that control, in turn, a fantastic number of RNA transcripts. So every protein that this bacterium makes, an RNA has to be made to make the protein. And that's the way it's regulated. It's a top-down system. It's really elegant, really beautiful, and very efficient, and very compact. But we can compare that to what's called the call graph in Linux, the Linux operating system. And what we see is that computer programmers, after millions upon millions upon millions of hours of code writing, because you know that we're all beta testers, right? You know that? They have come up with a system with a lot of modules that open up a huge number of submodules that control a few outputs. This is incredibly inefficient, but it's the best we can do. Now, if this is the best we can do with lots of intelligent design, does this look randomly evolved to you? Or is this maybe like hyper intelligently designed? Because I couldn't have thought of this. I couldn't program that. The best programmers in the world couldn't, because this is what they do. And there it is staring us in the face. A supremely regulated, beautiful architectural system with all the hallmarks of design. Now, the second dimension contains a lot of things: specificity factors, enhancers, repressors, activators, transcription factors, post-transcription regulation, alternate splicing. Oh, it's crazy. Let's talk about alternate splicing. Your body, you produce probably about 300,000 different proteins. Your eyeballs produce different proteins than your liver, right? It's about 300,000 different proteins. But you only have about 19,000 protein-coding genes. I thought it was one gene and one enzyme. I thought one gene made one protein, right? Isn't that what we've been taught since the 1940s and was shrined in law in the 1960s? Well, how does your body take 19,000 genes and make 300,000 proteins? It's something called alternate splicing. This is the wrong image for that. I don't know why I have this here. What happens is this. You have a gene, and the gene has sections in it. And when your body makes the RNA, it cuts some sections out, throws them away, takes other sections, duplicates them, rearranges a few sections, joins other sections together. It'd be like having a chocolate chip cookie recipe that's also a recipe for rocket fuel. By deleting lines or adding lines or joining lines together, you make something completely different. That's how we work. This is not something anyone anticipated, because it's a lot more complicated than anyone anticipated. And the reason for that is Darwinism has always taught us that life is simple. It's not simple. Life is incredibly complicated. I showed you this before. Oh, no, this is a different one. Here, this is alternate, okay, it's even more than alternate splicing. This is alternate reading. Here's a non-protein coding area of the genome. And that area, this polymerase, can make multiple different RNAs out of it. You see how they start and stop in different places? Those are different messages. So if you mutate that one letter, you might, honestly, on average, each letter in the genome is manufactured into 6 different RNA messages. One of them might affect how well you see in the dark, or whether you hiccup when you eat garlic, or how tall you are. Those have nothing to do with each other, do they? And so one change can affect radically different aspects of an individual. Natural selection breaks down in that system. Natural selection needs a really clear target. This skin color affects how much stomach acid I produce. What? It does. What? So if I have if I get indigestion or if I get sunburn, which is natural selection going to act upon? It can't. There's no clear signal, and that's what that biology has taught us. Oh, this is what I was talking about. Here's a splicing code. Here's a gene, a typical gene in your body. There's double-stranded DNA. The gene is only on one of the strands. Now, there can be a gene on the other strand going the other direction. Sometimes there's genes in between these spaces, too. That's all crazy complicated. But upstream of the gene, there's what's called an untranslated region. This is not made into protein. But this is where the activators and repressors attach. And they tell the cell, are you going to make an RNA of this or not? But once it makes the RNA out of the whole thing, it'll stop here. We have these red things. Those are the parts that code for protein. The blue parts, those are introns, intervening sequences. They don't code for proteins. They have to be cut out and thrown away, and then the red pieces joined together. But sometimes the blue thing contains an exon. Sometimes it doesn't. Sometimes it contains an entire different gene. Sometimes it's a piece of a gene going the other direction. And your cell looks at these things, and there's like little signals in between, these little sequences at the ends of each one of these things. And it will tell the cell, make this protein in this way at this stage of development under these conditions. So the same gene can make different proteins when you are growing in your mother, when you're going through puberty, and when you're almost dead. The same gene can make different proteins. Oh, so if a mutation happens there, which part of your life cycle is it going to affect? If it affects only you when like over 50 years old, basically, once you have children, you're not needed anymore. So dying of skin cancer, that's nothing that natural selection can touch because you've already had kids. Dying of a heart attack, doesn't matter. You're already old enough, you have kids, you've already reproduced. It's only things that affect reproduction that natural selection can touch. So things that affect childhood and adolescence and young adults. That's the only thing natural selection cares about. And there's so much complexity here that natural selection becomes blind. So therefore, Darwinism actually doesn't work. Okay, but that's the second dimension. We got two more. The third dimension of the genome is the three-dimensional architecture of the genome. I can't believe you're sitting here listening to me talk about this. Some of you sitting here, why on earth did I come here? If you look at a typical chromosome, there's some sections of the chromosome. There's what's called a centromere of highly repetitive DNA. Then the telomeres, or the ends of the chromosomes, also highly repetitive DNA, with very characteristic sequences. Then all the genes are in the arms, the short arm and the long arm. And that's basically its structure. Now, if you put that all into a cell, you think maybe they're just randomly jumbled around inside the cell? Because when we look at a microscope, they just seem to be randomly jumbled when the cell's about to divide. And the telomeres are actually anchored to the nuclear wall. They're not random. They're spatially oriented. And so if you have a big deletion or a big inversion or a duplication in your genome, it's going to affect how these things are organized in the nucleus. So if you have DNA, have you ever had one of those balsa wood airplanes? You know what I'm talking about? It has a rubber band, and you round up the thingy, and as you turn it, all of a sudden it goes kink. Because when it's full of twists, it can't twist anymore, so it kinks, and you keep on going, you get twists of twists. And you're like, yeah, I made this thing fly 100 feet. No, you break your rubber band. But here's the point. DNA is a double helix that does that. It is wrapped around proteins in your nucleus. They're called histones. And then like beads on a string, those wrap up to make a very compact structure. And eventually, they condense to the point where you can see it under a microscope. But this is twists of twists of twists of twists. And you can see, oh, look at that, fluorescent proteins. I love my PhD work. That's not my work, but that is amazing. They can now tag individual proteins with different colors so you can see them under a microscope. That's so cool. But when they start doing this, okay, that's at one part, that's not normal. That's only at one very short stage of the cell when the proteins get lined up and the cell divides. How is it normally when the cell's not going to divide? It's like this. Each chromosome has a defined place in the nucleus. It's not random, it's organized. When they first sequenced the human genome, one of the first papers I read afterwards, these scientists, they looked at genes that are used in biochemical pathways, like that lac-operon in E. coli. There's 3 genes right next to each other. They said, okay, in the human genome, this does that, and this does that, and this, and it's like 5 or 6 proteins in a row in a biochemical pathway. Let's go look for them in the genome. They predicted they'd be next to each other, because in the E. coli they are. So they said they're next to each other in the genome, and they looked, and they found out that genes that are used together are randomly spread out in the genome. And they said, oh, it's just junk. There's no rhyme or reason to it. The genes are just thrown in there and out of order. They're all over the place on different chromosomes. It's just millions of years of evolutionary accidents. Until they did this. And he said, oh, you know those genes that are on different chromosomes that are used they're used together? They're actually next to each other in 3D space. So whoever wrote the sequence of that chromosome knew that when it folded up into 3D shape, that this gene and another gene on a different chromosome will end up right next to each other. And not only that, genes that are used together tend to be in a pocket inside the nucleus that's right next to a nuclear pore. So when your genes need to turn on, they all turn on at the same time. An entire biochemical pathway turns on at the same time. The messenger RNAs go out the cell, and you can make all the proteins that you need all at the same time, all at once. Because the genome is actually incredibly efficient and incredibly not junky. So they were wrong. But if you bring this up online, they will pounce on you because you're threatening their edifice. Because if the genome is not full of junk, if life is not simple, then Darwinism is not possible. The fourth dimension is time. It turns out that the DNA sequence itself changes. Your liver cells have duplicated chromosomes. One great way to increase to increase your reaction rate is to give yourself more genes for doing some reaction. So your different liver cells have different chromosomes. They have different genomes. Your brain cells, when your brain is developing early on, see, if I made a brain, I would lay down a sheet of tissue and then make it fatter. That's not how your brain develops. There are nerve cell proliferation spots in the brain where the nerve cells are generated, and then they migrate through the brain as it develops and go to different places. And there are little pieces of DNA in your genome. They're called retrotransposons. They will circularize and pop out of the genome. Life is wonderful. But they will circularize and pop out of the genome, float around someone else, and stick into another place in the genome. And very often, these little pieces of DNA have something that turns the gene on. So if they're not there, the gene can't turn on. If they are there, they can turn on. And your brain cells, these little retrotransposons, they jump around in your brain cell genome, so your different brain cells have actually different genomes, and different genes turn on and turned off. What? So not only does, picture this. Imagine that you eat a bad peanut and you're detoxifying, the aflatoxins are gonna kill you. But your liver says, hey, I can get rid of that toxin. Now your kidneys don't do that, your eyeballs don't do that, your brain doesn't do that, but your liver says, I can get rid of that. I know where the gene is. And your liver finds the gene, twists around the chromosomes to expose that gene, makes a bunch of copies of that gene in terms of RNA, makes it into a protein, detoxifies that thing, and then when it's done, it folds the chromosomes up and packs it away again. That's the fourth dimension. That's a three-dimensional object changing shape in time. That is amazing. It happens over minutes. It happens during your lifetime because you don't need the same genes when you're a zygote as you do when you're going through puberty. I mean, you need different genes at different times. But there's so many other things. I won't even read the details because you get overwhelmed. And I could probably add 50 more lines to this if I wanted to. Ornish is a maverick scientist. A lot of people don't like him because he writes controversial things. But to my mind, he's amazing. This is a very, very interesting chart. What they did is they found a whole bunch of men who had enlarged prostate but didn't have prostate cancer. Because you're not actually allowed to experiment on human beings. You know that, right? You're not supposed to experiment on human beings. But because they didn't actually have cancer, they took these men, they took a biopsy, and on the left, they looked at what genes were turned on and what genes are turned off. Green is off, red is on. Above this line, these are happy genes. These are generic housekeeping genes that we express all the time in a happy cell. Below that are genes that are stress-related. So in these men, some of them are more healthy than others, fine. But in general, they had a lot of stress-related genes turned on, and a lot of the happy genes weren't activated as much. And here's what they did. They said, okay, come back in a couple of months. Follow these generic diet and these generic exercise regime. And they came back and they took another biopsy and ran the study again, and look what happened. The stress-related genes turned off, The healthy, happy genes turned on. We do not need any other evidence. We know exactly what controls human lifespan. Smoking will kill you. We know that. So will automobiles. Okay, fine. But people, not the super athletes, they don't tend to live a long time, but people who follow generic diet and exercise, light exercise, and a healthy diet, they're healthier on the inside. Okay, friends, you don't need any more information than that. You need nothing else. That's the secret to living healthy. Get your genes to turn on and turn off in the right proportions. Now, it doesn't mean you're going to live forever. No. But, okay, talking about changes in time again. We know that liver cells are different, brain cells. I said all that already. Okay, let's do this one. What is this? Go. Please go. Go for me because you like me? Go. Okay, is that what we showed you yesterday? No, because it hasn't had a TV around it. Oh, I have a version of it without the TV. Okay, cool. So for those of you who weren't here yesterday, we'll do this again. That is DNA. Let's turn that DNA into a protein. First, we have to make a copy of the protein, a copy of the DNA in a form of RNA using a very complex machine there. It's called a ribosome. The ribosome, we're showing it magically happen, but there's about 20 other proteins involved in this. It opens up the DNA, it lines up letter per letter on the DNA, and makes a copy of the DNA. So an A is copied into a T, and a G is copied into a C. Actually, a U, not a T. And now we have a piece of RNA. Now, if that was in you, you'd have to cut it up and resplice it. In bacteria, they don't do this. So maybe it's a bacterial RNA. And we're going to make that bacterial RNA into a protein using these transfer RNAs. On the bottom of these, they have a 3-letter code. And those 3 letters will match up to 3 letters on the RNA inside this other machine. This is called a ribosome. And it works about this fast. And it literally matches them up, shoves this thing in there, and off the top, that's the amino acids that pop off to make the growing amino acid or protein chain. That's amazing. This is happening in you right now, at least 1 billion times right now. These other proteins called chaperones come and glom onto that thing and prevent it from folding. And they will bring it to another gigantic molecule. It's a multi-protein molecule called a chaperonin. And this unfolded protein will go inside this watermelon-shaped chaperonin and get folded up into a protein. And what happened here is we started with a linear, one-dimensional DNA string with a 4-letter language where each word is 3 letters. And we're going to fold it up into a 3-dimensional, 20-letter language, machine, whatever you want to call that, called a protein. And that was the simplest version I could show you of this process. I mean, there's so many things we skipped over, it's shocking. You cannot have life before you have that protein. You cannot have that protein without the DNA. You can't get the protein without the copying systems that are made of proteins. You can't do any of this without ATP, that thing I showed you at the beginning, the ATP synthase motor. That was manufacturing the molecules that are used to power all of these things. But that machine's also made of protein. So what came first? The ATP synthase, the ATP, the code for these proteins in the DNA, or the machines that make the proteins that make the motor that makes the ATP that powers the machines. Yeah, good luck, Mr. Evolutionist. There's also this idea of junk DNA. Now, J.S. Maddock is a famous modern geneticist, and he said, the failure to recognize the full implications of this, particularly the possibility that now this is junk DNA, the intervening non-coding sequences, junk DNA, the stuff that doesn't code for protein. The failure to recognize the full implication of this, particularly the possibility that junk DNA may be transmitting parallel information in the form of RNA molecules, may well go down as one of the biggest mistakes in the history of molecular biology. I can just walk off the stage right now. The evolutionists were just wrong. We are much more complicated than you wanted to admit. So I submit to you, the genome is a multidimensional operating system for an ultra-complex biological computer. It has built-in error correction and self-modification codes. There are multiple overlapping DNA codes, RNA codes, and structural codes. There are DNA genes, there are RNA genes. It was incredibly well designed with a large amount of redundancy on purpose. Redundancy is a hallmark of design. When they launched the space shuttle, there were 3 primary computers on that thing. So if the first computer failed, they had a backup, and they had a backup to the backup. Anything that could be duplicated on the space shuttle was. What was not duplicated? The fuel tanks on the outside, the heat tiles underneath. Those are the failing points. But anytime they could duplicate something, they did, because that's what engineers do, and that's what we see in the genome. God used sound engineering principles during its construction. And yet, despite all that redundancy, it displays an incredible degree of compactness, because only about 20,000 protein coding genes create over probably about 300,000 different proteins. So I have a challenge for the evolutionists. This is a quote from Charles Darwin. Now, this has been very abused. I only use this with a caveat here. He said this, and then he followed up by trying to explain the origin of the eye. He said it keeps him awake at night, and then he tried to come up with this evolutionary thing. So he said this, and he thinks he can explain it, but he really can't. But he said this: if it could ever be demonstrated that any complex organ existed which cannot possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. Well, Mr. Darwin, welcome to the 21st century. Your theory has absolutely broken down. My retort is, I claim the genome could not have arisen through natural processes. And hey, Darwin, give us a workable scenario, including the source of the informational changes, the account of an amount of the mutation necessary, a description of selective forces. I'll even give you millions of years, because I know you can't do it. Once you have life, life can be modified. You can't get life from scratch. It requires a designer. Period. Now, again, a lot of this has appeared in the pages of Creation Magazine. I want to give you an opportunity now I want to explain to everyone at once so you know what's going on here. I would love for you to sign up for Creation Magazine. In fact, grandmas, moms, imagine your children's delight if they got a glossy science magazine in the mail that had their name on it. If I was that kid, oh man, I would tear that thing open and I would read. Now, a lot of people don't have print magazines anymore. Well, we have a digital version also. If you sign up for the print magazine, we will give you an email every time it comes out 4 times a year. There'll be a link in the email. Click on the email and it'll open up. I've read Creation Magazine flying across an ocean on my phone. So it works great. That's there for you. But for a 2-year subscription, I'm also going to include 2 documentaries. One is called Darwin, the Voyage That Shook the World. We spent about $1 million on this. For Charles Darwin's 200th birthday, we went all around the world chasing Charles Darwin's footsteps. And we interviewed a bunch of evolutionists. We went to Galapagos Islands. We went up and down South America. We went to his home in England. It's a really interesting documentary because you can't understand evolution without understanding that man. And that man is not the man that the media has portrayed. He wasn't some happy person. No, he was a miserable, suffering individual. In fact, he was agoraphobic. He would never go outside in a crowd. He had a mirror at the end of his older brother was a drug addict, so they pensioned him off, and Darwin inherited all the family money. Anyway, he'd be sitting in his study with his doors cracked, looking down the mansion's corridor, and there was a full-length mirror just inside the front door. And when the door opened, he could look up and see the mirror. If it was an unannounced visitor, he'd run away. Yeah, that's the man who started evolution. We need to understand him. I'm also going to throw in it's about a 35-minute documentary called Fallout. We went around to college campuses and interviewed hundreds of students and asked them, hey, first we asked them, did you go to church growing up? If they said yes, we said, all right, were you ever taught anything about creation or evolution? And then we said, do you still go to church? wow, they said the most interesting things. Take out all the swear words, and we had a really good documentary, and it's actually a genius production. It's only about 35 minutes long. It's quirky, it's fun, but you can get that with a 2-year subscription for the magazine. Here's what the sign-up form looks like. Check off 1 year or 2 year. Those will be going around. They'll probably be going around while we're doing a Q&A after this also. But just look at it. On the back is a place you can give a gift subscription to somebody. Now, on the table, the high-tech cell, very similar to what I just presented to you if you want to see it again or share it with somebody. But also, because a lot of people don't have DVD players, the DVD has a QR code on it, and any modern phone, if you hover over it, it'll bink, and you click on it, and it'll go straight to CMI's web store, and it will load that for free into your account. And so you can watch it. And after you do that, give the DVD to somebody else. Fine enough. That's there. That works great. Evolution's Achilles heel, the book, the documentary. We're talking about high, powerful stuff here. A lot of the things we do are like on the 8th grade level, and we have a bunch of kids' books, but this is like upper high school, college level on purpose. Sorry, guys, I forgot to say, yes, you can do that. Appreciate that. So these materials are there for you. And the reason I brought the books and things is because I'm going to go away. I can't tell you everything. I can't tell you everything I know. You wouldn't want to know everything I know. You're interested in other things. There's a huge selection of things answering tons of questions, and we believe they're faith-affirming things, and they're there for you. And as we break into Q&A time, I'm going to leave up 1 Peter 3:15. But in your hearts, honor Christ the Lord as holy, always being prepared to make a defense to anyone who asks you, for a reason for the hope that's in you, yet do it with gentleness and respect. Why gentle and why respectful? Because trying to emulate Christ, it's hard, man. When I get insulted, my Irish temper flares up. I want to punch someone in the nose, but I can't do that. I'm not allowed in Christ to have that reaction, so I got to suck it up and give an answer. Now, I don't expect you to do that, but okay. I have 8:03 on my clock. I like doing short Q&As because people get antsy. So, who in the front row here has a watch? Anybody? Oh, yes? Yes? Okay. At 8:15, do this. All right? Okay. Just like last night, I'll try to keep my answers short. I will try to answer as many as I can. I'm going to try to answer it and then point you to something that might answer it more fully, a book or a DVD or a web article or something. But can we avoid theological questions? I don't want to be tempted to contradict something that the organizer of this thing might believe. That wouldn't be fair. So nothing about head covering or Bible translations or end times theology or free will versus predestination or no. But anything about science is fair game. I'll just try to answer it as best I can. Okay? I don't promise I know the answer. I'm really putting myself out on a limb here. Who wants to start us off?
What are some problems that creation scientists are having trouble solving today?
What are some of the problems creation scientists are
Trying to solve today, especially that evolutionists might take advantage of in an argument.
okay, our weakest points are starlight and time.
But you said that's not a weak point because evolution.
Well, no, they also have a weak point. It's a two-coca argument. Oh yeah, well your theory's bad too. That's a logical fallacy. But they have observational evidence that makes the universe look billions of years old. They just can't explain where it came from. We have reasons for believing that observational evidence has been distorted by time ticking at different rates. But I'm uncomfortable with all of them, honestly. That's my hardest one. Next, after that, I'd probably say radiometric dating in general, but not carbon dating. That's ours. But when you look at rocks and things, you can radiometric date, and the older rocks have older radiometric ages than the rocks on top. Now, part of that is because there's probably a burst of accelerated radiometric decay during the flood that has been modeled by physicists. There's very good evidence for that. But then again, that's way outside my field too. So one reason I feel uncomfortable in those areas, because that's not my expertise. Let's talk about DNA. I'm going to pound you over the head with genetics. Radiometric dating, not so much. Sir?
I'm not even sure how to ask the question.
Okay.
What in the second dimension, I forget what you call it, where the cells are going through and exons and are splitting and throwing out things, what controls
What controls the splicing and dicing of the genes? Things we're just learning. Basically, it's the non-coding RNA makes a massive amount, the non-coding DNA makes a massive amount of RNA. That's where all the decisions are. So if we were a computer, the read-only memory, the ROM, is our DNA. The DNA is made into RNA, which I would call random access memory, RAM. That's where all the changes happen. That's where all the decision-making happens. Massive amounts of things are happening at the level of RNA, and then a small amount of all that decision-making results in proteins. The what? The hard drive is the DNA. The DNA is stored in the hard drive. So it's the non-coding RNA where all this decision-making is happening. And it affects how chromosomes fold up. It affects where they're located. It affects what's sticking on the chromosomes that allows genes to turn on and turn off. There's a lot of rheostats in the cell where it'll be fine, and all of a sudden it's like, oh, I'm going to make more of this, but okay. And there's some actually switches where things turn on, turn off. And it's really, 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 really complicated. And all I can say is go get a college textbook and start reading. Or don't go online because there's some good information online, but a lot of it's just really ridiculous. It's hard to find a good source of good, solid information online because people post a lot of pretty low quality stuff. I'll just leave it at that. We're only open. It's like Pandora's box for the evolutionist. We've only opened it up. We haven't dove in yet. It's not metaphysics. It's not vitalism. The cell doesn't have a life force that's controlling things. It's very mechanical, especially for bacteria. But it's just hyper-complex mechanical. Notice I didn't answer the question, so because it's not answerable.
Obviously, the proteins are involved here, but there's chemicals we talked about, complex chemicals. Really, what I'm after is what is telling all this stuff that needs to happen.
Really, I can't answer the question. Because every single thing has its own control system and its own own set of variables. And the cell will actually put methyls on all this DNA and turn off genes, just methylate it. That gene can't be made into a protein. And it'll modify the tails and the histones like 20 and 30 times for each tail. And that tells all this information in this very complex system. I'd have to give an entire talk on just that. Okay. Peanut gallery? Right side? Sir?
Yeah, how do evolutionists explain the patterns that you showed when they sequenced the human genome?
How do evolutionists explain patterns in the human genome? Like when I showed, where is it? When I showed that, they call that junk DNA. That's a highly repetitive, this is just ACGCC, 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 millions of times over. And honestly, the number of ACCGCCs or whatever it is isn't important. You could delete huge sections of that and your body would still do just fine. So it's not intrinsically important, but that spacer region with no genes in it affects how the DNA bends. It affects where the genes are located in 3D space. So it's not like every single letter has to be coding for something. Some of it's just scaffolding. And just like a building, you can knock out some of these things. A building is going to stay up, except you wouldn't want to be here in a tornado or under heavy snow load. But except in extreme things, you could. I mean, this building is over-designed. Why? Because we don't like ceilings that collapse and kill us. We make them much stronger than they need to be. Same thing in the cell. You can delete a lot. You can't delete it all. In fact, they've done a lot of deletion experiments in a lot of different species. Take out this, take out that, take out that. And either take out genes to see which ones are absolutely critical for health, or take out spacers, and they find out that there's a certain minimum. You can't go below that, or the cell just dies. So it's important. It's not 100% necessary for survival today. We're going to talk more about that on Monday night. Sir.
Yeah, we're talking about DNA and stuff. What do you think about Christians having part of their DNA and stuff, or anyone having part of their DNA taken out because of curing cancers and stuff?
What do I think about humans that have genetic modifications? I have a friend who is dying of Huntington's disease. She was one of the most beautiful young girls I've ever known. She was a total sweetheart. She's now incredibly mean, totally conspiratorial, and her body's wasting away. Her father died of it, her brother died of it. She has 2 beautiful children and a broken-hearted husband. If she could be fixed, and those little girls could have their mom back, and she wasn't a witch, because it melts your brain down, would I say no? The problem with genetic engineering is not humans, adults, it's will you modify the germline? If an adult says, I'm blind, I can get an injection, and the genes can fix me and I can see again, would I say no? That would be a wonderful thing. But if that DNA gets into the reproductive cells, it can have off-target effects. So if you're an adult, you have plausible deniability. What's it called? When you can consent to something, informed consent, but a baby can't, and an unborn baby certainly can't. So we have to separate adult consenting human from baby. It's not fair to potentially affect a baby when genetic modification. Most people in history we tried that on are dead because it didn't work. But they can now they'll take a sample of your own bone marrow, give you leukemia treatments to kill off all your bone marrow, genetically modify that and stick it back in your bones, and all of a sudden you don't have sickle cell anemia anymore. That's amazing. The real problem, though, is, hey, doctor, how did we learn that information? Oh, yeah, we experimented on fetal cells to do it. That's the problem. It's the abortion and the fertility clinic industry. They make a lot of money selling fetuses to laboratories. And a lot of medicine is performed on those things. So I know it's creepy, but if we're saving or prolonging or improving human lives, that's a good thing. And when Jesus was healing people blessed him for it, right? It's just that humans are evil and everything we do is tainted with sin, and it's hard to separate one industry from the other. And it makes me sick even to think about it. Okay, politics. I'm not supposed to talk about politics. When George W. Bush, he made a command for the government, no new lines. So we can't go back in time to the 1970s where they did something horrible and now we have a fetal stem cell. I don't want to use the word out loud because there's children here. But it is something horrible. They made a stem cell lineage from a embryonic kidney, which means it wasn't just a little ball of cells, it already had kidneys. And they made a line, HEK 293. It's been used to test every medicine you've ever had. It's been used to test all the food additives in every food you buy at the supermarket. Every colorant of every fabric you wear. HEK 293 is like an industry standard. And yeah. He said no new lines. And the industry went ballistic because they didn't have any African American lines. European genetics is different. So if we're going to treat disease in African Americans like hypertension and things that are really common, we need African Americans or African cells. And so Harvard raised over $1 billion of private money. And so did Caltech, and so did a lot of other universities. They did private stem cells the government has no control over. And so good intention, but because people are evil, they said, hey, everyone, give us lots of money, and all of a sudden we don't need government grants. We got all this money. We can pay our researchers to do anything they want. Now, right now, it's illegal to raise an embryonic cell past the 16-day window. Because at 16 days, that's when the embryo starts developing a backbone, or at least a spinal cord. And it's 16 days is the last time when twinning can happen. So identical twins can happen up to 16 days. The embryo can split in half. And strangely, at the last minute, you tend to get a left-handed and a right-handed twin. They're identical. And since they say it happens all by itself up to 16 days, that's their window right now. There are a lot of people pushing against that because they want to do experiments later on. And that becomes really gross really fast. But it's 8:17. Have you been doing that? I've not been looking at you because I'm trying to answer this one question. I don't want to end on a downer. I know that's very disturbing, but welcome to science. Science is amazing. Look at the experiment we did today. I can't believe that actually worked. We should rejoice in this world that God has created us, but we also have to walk wisely, very wisely. And we have to be informed on the big ideas. And we can't have a knee-jerk reaction against, climate change. I know the communists are pushing climate change. I know there's a political agenda behind it. But it doesn't mean that it's not true. It should be a scientific issue, not a political issue. But when you get politics and science mixed together, you never get good science. That's a huge issue and is extremely frustrating. The whole debate right now on abortion, the government blew it. The Supreme Court gave it to the states. Why? So the states can be evil? No, Supreme Court, you should say murder is wrong. It's illegal. But they didn't. They punted. And that was the conservative justices, Amy Coney Barrett, the Catholic anti-abortion justice, right? Now it's a state issue. Now half the states have legalized it, even New York, I think, up to birth. That's called murder. But this is science. It all gets intertwined into this big, ugly mess. And I'm getting negative again. I'm trying to think of something positive to say so we can smile as we walk out of here. All right, do we have announcements to make? I'm going to end in a prayer. Are we going to? All right, let's pray, because we got to pray out of this mess we're in right now. Father, thank you for this beautiful, beautiful, beautiful world. Thank you for the amazing things that you put inside of us and inside of other living things. Thank you, Father, that you've just allowed us just peek in just a little bit, and we're stunned, and it's shocking, and it's just beautiful, and it's so complicated, and life is just screaming a testimony up to the heavens saying, God made this. Help us to accept that. Help us to believe it, and Lord, help us to be able to figure out ways to share this with other people. Because if people could see that life could not have evolved because it's too complicated, then maybe, just maybe, they'll be more willing to believe in you and your word and the Bible. But Lord, help us to be wise. Help us to learn and not become tainted by the knowledge. Help us to act upon the knowledge that we have. And help us, Lord, to not forget that you are the Creator and you love us no matter what. Thank you this evening. Bless us to sleep well tonight. Amen. Thank you.
Amen. Thank you for that, brother. Wow, just think about Psalm 139 again. For you formed me in my inward parts. You knitted me together in my mother's womb. I praise you, for I am fearfully and wonderfully made. Wonderful are your works. My soul knows it very well. And I like how he asked, does my soul know that? And so as you hear things, as I hear things that go way over my head, that helps me understand that we serve a creator God that is way more brilliant than us. So thank you for that. And it's exciting to hear. Again, science cannot make moral decisions. So we need God's word. We need God's word. And just love to have Dr. Carter explain a little bit more some of his work he alluded to yesterday of taking colored proteins and making fish different colors and things. So he's done some incredible work himself. So thank you, brother. Just a couple announcements. The service tomorrow morning will be at 10 o'clock in here. We'll have a Sunday school hour. Brother Keith Bucher will be leading that, and then Edgar Kinsinger will be having the message tomorrow. So that's here again at 10 o'clock tomorrow morning. And then 5 o'clock Dr. Carter will be at Providence Mennonite in the morning, and then in the evening at 5 o'clock at Trinity Mennonite in Odin. So he'll be speaking on the alternative, creation's competitive edge. So you'll want to go listen to him there. And I think both morning and evening it's the same presentation. So yeah, what a blessing to be here. There is some offering boxes that will help cover the expenses for the weekend. And also there is snack provided for everyone here at 8:30, so in a couple minutes, hot dogs and ice cream, so everyone's invited to that. And also tomorrow, after the service, there will be a meal here, and everyone is invited for that. It's free of charge, so you're welcome to come to that. So again, Dr. Carter, thank you for giving us a little bit of God's awesome creation, and that should lead us to worship him. Well, how amazing that this same Creator God that is way, way above us has stooped to come to this earth and live here among us and die for us, that we can know him and someday live with him forever. It's amazing. All right, you may be dismissed for snack in the dining area.